Namaste students. I am Javin Dr. Aziz Mohammad Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapith Women's University, Jaipur. Today I am going to discuss about the topic single chain antibodies and domain antibodies. So, in previous video we discussed about the antibodies and the classification of antibodies, what is variable region, what is the concentration, and monoclonal antibodies, and also the genetics of immunoglobulins also will be discussed here. So single chain antibodies are also known as the single chain variable regions. So we know that each antibody have a variable region and a constant region. So variable region is a region which is different in each antibody. And when we talk about the construction of an antibody in the in vitro conditions, then we can say that a variable portion is used for the variability, and that variable portion is sufficient for the construction of a whole antibody. So the difference is only in the concentration. No differences in the variable regions. Variable region is difference is only in the variable region. Concentration is equal in each antibody, and we can say that concentration is found in the in vivo conditions, and for in vitro conditions we can take the use of the variable region, and that variable region is sufficient for the construction of the whole antibody. So a single chain variable fragment is not a fragment of antibody, but it is a fusion protein of the variable region of the heavy chain and the light chain. So if we talk about a mm, complete antibody, it is a variable region in the heavy chain and the light chain. So in the variable region, there is a heavy chain and the light chain. So if we talk about the single chain, in the single chain, a heavy chain is there and a light chain is there. And the heavy chain and light chain are connected with a short linker peptide to 10 to 25 amino acids, and the linker is usually rich in the glycine for flexibility, as well for the serine and threonine are used for the solubility. The VL and VH are used for the construction of a single chain or single chain variable fragment, and these molecules are created to facilitate the phase display where it is highly convenient to express the antigen binding domain at a single peptide. As alternative, single chain variable fragment can be created directly from the subclonal heavy and light chain by the hybridoma technique. If we combine the heavy chain and the light chain in using the hybridoma technology, then we can create the single chain variable fragments, and these are also considered as the monoclonal antibodies. Some another technology can also be applied, such as the flow cytometry, not chemistry, for the construction of the single chain variable fragments. So how these single chain variable fragments are purified? As we know that a single chain variable fragment do not have any concentration, so it will not have any kind of FC portion. Okay, it is not a complete antibody; it is only the variable fragments, which is it contains heavy chain and the light chain, so it do not have FC portion. And so the common binding protein G, cannot be used to purify the antibodies. The variable fragment can be purified by the protein L. Okay, it can be purified from the protein L, since protein L interacts with the variable region of the kappa light chains. This variable L chain protein L will bind to the variable region of the light chain, and they can be six histamine proteins also. So by using the protein A and the protein L, we can purify a single chain variable domain. Now talk about the bivalent and trivalent single chain variable fragments. If we talk about the one variable fragment, then VH, one VH and one VL, one VH and one VL will combine to one another VH and VL to make a di single chain variable fragment, and they will make a di body. And when one die body will combine one another single chain variable fragment, they will create one tri single chain variable fragment or tri body. Let me explain it with a diagram. Suppose this is a single chain variable fragment. This is the VH region and the VL region. This is the single chain variable domain. Suppose this variable domain will interact to one another variable domain, then it will make a tandem die single chain variable fragments. So if they both of them will combine to each other, they will make a die body. So this is flanked by VL and VL and VH and VL, which will make a die body. When this die body will bind to another single chain variable uh, fragment, then they will create tribody, means three single chain variable fragment will mix together to make a tribody. So what are the examples of the single chain variable fragments? Axial jump is a single chain variable fragment, and they will bind to the complement system, and Rolu jump is another single chain variable fragment which have the binding to VGF-A and used to treat the weight age-related muscular degradation. Similarly, C6.5 is also a diabody which has good capacity to target HER2 new, which is found in the some breast cancers. Now we talk about the single domain antibody. Single domain antibody are the smallest structure of a antibody. Means the smallest part of the antibody is known as single domain antibody, which has the capacity to bind to any kind of antigen. Okay, in the single domain, single chain antibody, there was a variable region and the heavy chain VL and VH region. But in the single domain antibody, it contains only the variable portion of the either heavy chain or light chain. Only VH or VL will be there. Heavy chain only antibodies. They are known as the heavy chain only antibodies. If only VH region is there and if only VL region is there, then they will be called the light chain only antibodies. So how they are created? They are synthesized by the on isolation of the either isolation of VH region or VL region. So only that VH region and the VL region will be sufficient for the isolation, for the identification of the complete antigen molecule. So here we can understand this with the diagram. So this is a V domain of a amino acid sequence. Here VH region is the heavy chain, variable region of the heavy chain is there, and this is the variable domain where VH portion is there, and here this is the immunoglobulin where this is the VH and then this is VL. So only this VH, either this VH or this VL region will be used for the construction of a complete immunoglobulin. Here this is the IgG2, IgG3 molecule, and from this IgG2 and IgG3, this portion will be used for the construction of a complete antibody, and this can be used for the isolation and identification of a complete antigen also. So only this portion is sufficient to act like a antibody, and they will be isolated, they will be purified using particular enzymes, particular amino acids by enzymatic methods. So in this video, we learned about the single chain antibody and the domain antibodies, which are the smallest structures of the complete antibody, which can be used for the isolation, for the construction, for the synthesis of a complete antibody, and for the binding of a and binding to an antigen. So this smallest portion is enough to bind to a complete antigen. And what kind of questions can be asked in using this lecture? One can be asked that what are the single chain antibody and single chain variable fragments? And the second questions can be asked that what are the domain antibodies? So my lecture is over here. Thank you very much.